All right, hey nerds, we are here to talk about the learning targets for Unit 6, which is on gene expression and regulation. This is maybe our largest unit, and so I'm a little concerned that this learning targets video is going to get too long. So I'm going to for sure make it through the first 6, and then maybe I'll pause the video and then make another video for the last 5. We'll see how the timing goes on this. I just don't want it to get too long, so I just got to keep track of the time. Okay, let's start digging in. It says here, the first one says, describe the structures involved in passing hereditary information from one generation to the next. So that's talking about the shape of the DNA molecule. DNA, and in some cases RNA, is the primary source of heritable information. Genetic information is transmitted from one generation to the next through DNA or RNA. It's stored in and passed to subsequent generations through DNA molecules. Okay. Prokaryotic organisms typically have circular chromosomes. You definitely need to know that. While eukaryotic organisms typically have multiple linear chromosomes. So we say that our 46 chromosomes are linear, but the main chromosome of a prokaryote is circular. Now, what is easy to mix up there is letter C. Prokaryotes, usually we don't hear about this in eukaryotes, but prokaryotes can contain plasmids, which are small circular pieces of DNA. And those are the ones that get that they get copied and passed during conjugation. So just be aware of that. That's not as common in eukaryotic DNA as it is in prokaryotic DNA, but lots of bacteria have plasmids, and those plasmids end up being super important in genetic engineering experiments and in transformation. All right, moving on. Describe the characteristics of DNA that allow it to be used as hereditary material. DNA and sometimes RNA exhibits specific nucleotide base pairing. So you absolutely need to know that A is across from T. That's in DNA. But then in RNA, it's A across from U. So please, 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 please don't make this really low-level mistake. If you see a pattern of A's, T's, C's, and G's, so it's like ATT, C, G, G, ATT, it is automatically DNA because it has T's in it. But if you see that almost identical pattern, but this time it says AUUUTCG, oops, I said a T, AUUCGGUUAAUGCC, that's automatically RNA. So just be aware of that. Pay attention. If there's U's in it, it's RNA. If there's T's in it, it's DNA. Okay, and then C is always across from G. There are the DNA molecule always has the same size. And that is because there are molecules that are purines that have a double ring structure, and there are molecules that are pyrimidines that have a single ring structure, and they're always opposite each other. And why don't I just quickly find that one? Nope. Yeah, this is the one. Show you that really quickly. So here are the pyrimidines and the purines over here. Notice the double ring structure there and the single ring structure, and then a double ring structure and then a single ring structure. So that helps DNA always maintain the backbone is always a certain distance apart. Notice that there are, just noticing this, there are hydrogen bonds in between the nitrogen bases. So those are weak, and so the DNA sides of the DNA are easily split when we need to do replication or transcription. But then the backbone of the DNA, let me see if I can scooch up a little bit. It's like everything is frozen. There we go. I don't know why I all froze for a second. Okay, so the backbone of the DNA, which is the phosphate sugar, those are all covalent bonds. Those are called phosphodiester bonds. They're super strong. So the backbone of DNA and the backbone of RNA never come apart. I mean, pretty much never come apart. Whereas these hydrogen bonds, super weak, and so they can pull apart very easily. All right, getting back to our learning targets. I hope my computer is not going to be too glitchy. I'm going to be super frustrated. All right, and I will tell you my little hint of how I remember which molecules are pyrimidines and which are purines. Pyrimidine, the word pyrimidine has a Y in it. Cytosine and thymine both have Ys in them as well, and they are pyrimidines. Pyrimidines have a single ring structure. I don't know, and uracil doesn't have a Y in it, but T and U are interchangeable, sort of, so I'm sure that didn't help a whole lot. All right, number 3, describe the mechanisms by which genetic information is copied for transmission between generations. So the first thing we want to talk about quickly is replication. And let me make sure I can go to this one quickly, and we'll just walk you through replication. This one over here. So just reminding you, let's see if we can find the most important parts. So initiation, yeah, good enough. Okay, so we start with an enzyme that's called topoisomerase, and topoisomerase is kind of untwisting the supercoiled. It's untwisting that. Then you have helicase, which is also untwisting and unzipping, so it's separating the two sides from each other. Then we have primase, which initiates the replication, in this case, by laying down an RNA primer first. You guys made a video about this, so hopefully this all rings a bell. And then we bring in DNA polymerase. So these are the enzymes that you are required to know. You need to know topoisomerase, primase, helicase, DNA polymerase, and then the last one you need to know is ligase, and we'll get to that in just a second. So DNA polymerase is going to read in the 3' to 5' direction, and it's going to build in the 5' to 3' direction. On the leading strand, that happens continuously. On the lagging strand, because it is anti-parallel, which means it's running the opposite direction, DNA polymerase has to copy in this direction. And so as helicase is opening more and more of the replication bubble, new DNA polymerases have to hop on and fill in the gaps. And those sections of DNA, those are called Okazaki fragments. Let me see if I can find you a good image of that. Those fragments are called Okazaki fragments, and these are these blue strands right here. And then they have to be joined together, because notice there's a gap between the Okazaki fragments here. Remember, I told you those were covalent bonds, phosphodiester bonds, and they're really strong. But when they were made separately, they're not joined. And so ligase is the last of the enzymes that you're required to know, and it creates that covalent bond between them so that they are one solid strand of DNA. And then we learned from the Missense-Stahl experiment that the way that it's being made is called semi-conservative, which just means that you have an original DNA molecule, it splits, and then each side is copied. So you have an original side and you have a new side, and that's the semi-conservative model. All right, let's head back to the learning targets and see if there's anything we didn't talk about. DNA is synthesized 5' to 3'. It is semi-conservative. Helicase, whoops, I'm trying to just get it to move. There we go. Helicase unwinds the DNA strands. It also separates the DNA strands. Topoisomerase is up front, really doing the supercoiling and uncoiling the supercoiling. DNA polymerase requires DNA primers. So that's with primase. That's the enzyme that does that. DNA polymerase synthesizes new strand of DNA continuously on the leading and discontinuously, making Okazaki fragments on the lagging strand, and then ligase joins those fragments. And that is it for this section. All right, next, describe the mechanisms by which genetic information flows from DNA to RNA to protein. So that right there, DNA to RNA to protein, that's called the central dogma, and that's this prezi here. If I back way out, you can see that the entire prezi is called the central dogma. That's what we're referring to, is this idea that information flows from DNA to RNA to protein. Now, the one exception to that we learned about were retroviruses. Retroviruses have RNA as their primary information source. Then they have an enzyme called reverse transcriptase that converts the RNA into DNA, and then the DNA into RNA, so it kind of goes back and forth, and then over to a protein. We'll talk a little bit more about that in an upcoming learning target. All right. The sequence of the RNA bases together with the structure of the RNA molecule determines the RNA function. So remember that mRNA is single-stranded. tRNA, the trucks, those are folded up on each other, and they form, they call it a hairpin structure with an anticodon down at the bottom, and it carries the amino acid up at the top. rRNA, that's the ribosomal RNA, so it's really all bound up and makes the ribosomes. And then we even learned about small RNAs that are like little double-stranded RNAs that lead to the degradation of other mRNA molecules. We'll talk about those more in a little bit, but the structure really determines their function. So here we have mRNA molecules carry information from DNA to the ribosome. That's why they're called messengers. Distinct trucks, tRNA molecules, bind specific amino acids. So each truck is defined by its anti-codon. And notice the T, anti-codon for the tRNA. So that's the opposite three letters of the codon on the mRNA. So each truck carries a very specific amino acid. However, the idea of wobble is that there are several trucks that carry that one amino acid, but any one truck will never carry a different amino acid. So that's the idea, that it is redundant. Several trucks will carry it, several tRNAs will carry it, but it is not ambiguous, which means you don't sit there and wonder which amino acid is it going to bring up. It's going to bring exactly the right amino acid every time. All right, tRNA is recruited to the ribosome during translation. Remember the difference between the words transcription and translation. I hate those words too. So transcription is making RNA. Translation is making the protein. Does that help? No idea. All right, so tRNA, the trucks come to the ribosome during translation to generate the primary peptide sequence, aka we're making a protein here. We're putting a whole bunch of amino acids together. So that's a polypeptide, also called the primary peptide sequence, also called a protein, also called an enzyme. Oh my gosh, so many synonyms. All right, our RNA molecules are functional building blocks of the ribosome. So good enough. I keep highlighting things and I don't mean to be doing that. Genetic information flows from a sequence of nucleotides in DNA to mRNA bases to amino acids in a protein. We already got that. I'm on C. RNA polymerases use a single template strand. Ooh, this is one that we're going to have to review a little bit. A single template strand of DNA to direct the inclusion of bases in the newly formed RNA molecule. The process is known as transcription. So when we're making RNA, we're copying only one side of the DNA. So look at letter D, because I don't think these words have come up in the prezzies. Letter D says the DNA strand, so the one side of the DNA that's acting as the template for the mRNA, is referred to as the non-coding strand. I think that's confusing because I want to call it the coding strand. So the non-coding strand, the minus strand, or the anti-sense strand. What? That's craziness. Selection of which DNA strand serves as the template strand depends on the gene being transcribed. All right, letter E. Oh, I wanted to go over transcription here with you really quickly. Let's just remind you about transcription and how that happens. And let's do, so it's got an initiation, elongation, and termination section also. So initiation, we've got the TATA box in the promoter. So TA, lots of T's and lots of A's, upstream from the gene that we're going to be transcribing. So that's where the RNA polymerase sits, is on the promoter. Yeah, and then in eukaryotes, RNA polymerase requires the transcription initiation factor or complex. Sorry, that includes all of these transcription factors. We come across them again later in the eukaryotic gene regulation notes. We see these little purple proteins. There's some darker purple proteins that get introduced in that section. Promoters have a characteristic DNA sequence, the TATA box, that comes at the beginning of the promoter. All right, and then elongation. Let's see, how do I get down there? Elongation, similar to DNA replication. Come on, go faster. RNA production occurs in a 5' to 3'. The template strand is the one that the RNA transcript is being produced off of. So we called that several different names: antisense, non-coding, I can't remember the other ones. The non-template strand or coding strand of the DNA will have the same sequence as the RNA transcript. Don't ask, it's just because they're flipped around, they're opposite letters. Okay, and then the last section is termination, and we don't need to worry about termination right now. So that's the heart of transcription. Let's see if there's anything we don't know. I highlighted those names for the template strand just because they might not be familiar to you, and I want to make sure you don't stumble on them and then not know what you're doing. The enzyme RNA polymerase synthesizes mRNA 5' to 3', reading the DNA 3' to 5'. Okay, so then we have the immature or pre-mRNA. Let's get out of here. We have pre-mRNA. Skip all this. Trying to find... There we go. So we have the pre-mRNA that needs to be modified. And so there are three important things that we do to make them mature. This is mature mRNA, and I can tell because it has the 5' cap, it has the poly A tail. These two, the cap and the tail, appear to allow the mRNA easier access out of the nucleus through the nuclear pores. They can get through more easily. The poly A tail also appears to determine how long that mRNA can live. There are enzymes that eat it from the back end and recycle the parts. And so the longer the poly A tail is, the longer it takes those exonucleases to eat and recycle the mRNA. So that increases its time frame, its lifespan. And then we have to cut out the introns, and I think that that's coming up here. So the introns are not coding. They're not coding for DNA. They do other things, but they're not coding for the DNA. So we splice out the introns and put the exons together. This is only in eukaryotic RNA. There are no introns in prokaryotic DNA or RNA. So that's kind of an interesting difference. Okay, that should be good enough. Let's go back to our learning targets and see how we're doing here. Introns and exons, and the idea of putting the exons together in alternate orders or putting in different introns, that's called alternative splicing. And that means that one gene can code for more than one protein, not a million proteins, but more than one protein. All right, number 5 is where it starts to get a little very repetitive, and I just want to make sure you're feeling clear about this. Phenotype, when we first learn the word phenotype, I always say it's what it physically looks like. Phenotype is the gene expression. It is whatever the proteins that are being made, that's the phenotype. So for example, it could be what I physically look like if my phenotype is brown eyes and the protein that's being expressed is got a lot of melanin in it and reflects light in a way that makes my eyes look brown. That's the phenotype. But it's really talking about what protein is being made in my eye that makes my eyes have the appearance being brown. My eyes are green, they're not brown. I don't know why I said brown. But anyway, so that's determined, the way my genes, the way my proteins are being expressed is determined by the genes that I inherited from my parents. And we call those the genotype, whether I got a dominant or a recessive, et cetera, et cetera. Okay, translation of the mRNA to generate, so translation is making the protein synthesis of the mRNA to generate a polypeptide, a protein, occurs on ribosomes that are present in the cytoplasm of both prokaryotic and eukaryotic. And I want you to just remember that prokaryotes, let's see if I can get out of here, prokaryotes, because they don't have a nucleus, they can do everything simultaneously. So they can do transcription and translation simultaneously. I'm trying to find the image where it shows that. I think that's down here, isn't it? Yeah. So this is the bacterial DNA. So this is their circular chromosome. These little things that I call guitar picks, those are the RNA polymerases. So they are transcribing the DNA, making RNA. That's this red line. And the ribosomes, a whole bunch of them are snapping on at one time, and they're making the proteins. They're translating the mRNA as fast as the mRNA is being produced. So we say that transcription and translation are coupled in a prokaryote. They're happening simultaneously. That is not the case in a eukaryote. So just a couple other things. Let's mention the genetic code here really quickly. You need to be able to read a table that looks like this. This table is fairly easy. I'm not sure what the table will look like on the AP exam. In this case, you see the codes themselves, and then you see what they code for, the amino acid that they code for. In the table that I showed you guys, the first letter was on the left, the second letter is across the top, and the third letter is across the right. And that's what this is also. But you need to be able to read it that way because not every table gives you the codons themselves. So notice that there are 3 stop codons that code for no amino acid. There is only one start codon. Every single mRNA starts AUG, and it carries the amino acid methionine. That is all I can think of about that one. Okay, so let's take a look at how it goes through. Let's remember the different parts of the ribosome. So as the mRNA is pulling through, there are 3 locations in the ribosome itself: the A location, the P location, and the E location. A is where the very first truck pulls up, and it has its amino acids. So we'll show it right there. This is the truck with the opposite 3 letters, the anticodon, and its amino acid. Now the other truck that is already there, and here's the growing polypeptide, this polypeptide now makes a bond with the, peptide bond. Oh my gosh, I had trouble remembering that name. It makes a peptide bond with this amino acid here. And now this truck can pull out and leave through the exit. And the ribosome is reading 3 letters at a time. It ratchets 3 letters at a time. And this is why frameshift mutations are a problem, because frameshift mutations screw up that grouping of 3. Okay, I think that's about everything we need to know about that. Trying to see if there's anything else you need to know. Elongation, termination, protein synthesis, eukaryotes. Oh, one other thing that I do want you to be aware of. Eukaryotes are so big that many of the proteins need to be made on the endoplasmic reticulum so they can be packaged and transported to other areas of the cell. And so in that case, when they are first coming together, when the polypeptide is first being made, there's a signal set of amino acids at the beginning that signal a protein called the SRP protein. That protein gloms onto the protein that's being built and drags it over to the endoplasmic reticulum so that the entire polypeptide or protein gets made within the endoplasmic reticulum. That's what gives the ER its rough look. So the rough ER, now it can be packaged and transported to other areas of the cell. All right, so let's keep going and take a look at how we're doing here. Initiation, elongation, and termination. Translation is initiated when RNA in the ribosome interacts with the mRNA at the start codon. Triplets are called codons, genetic code chart. Nearly all living organisms use the same genetic code, which is evidence for the common ancestry of living organisms. So that's evidence for evolution. tRNA brings the correct amino acid to the correct place. The amino acid is transferred to the growing polypeptide chain, and the process continues along the mRNA until a stop codon is reached. Okay, genetic information, retroviruses. This was one that we had to, this set of notes that we had to include all the way at the end. It doesn't fit very well in this particular unit, but retroviruses, remember, the DNA that they have inside them. So all of viruses, you guys, is DNA surrounded by protein. That's all it is. It's not made of cells. It's smaller than a cell. And so we don't consider them as alive, at least at this time in history, we don't call them alive. So retroviruses, instead of having DNA, they have RNA. And what that means is the RNA is the hereditary information. It's converted into DNA by the virus or by the enzyme called reverse transcriptase, which is then converted into RNA and then into the protein. The problem for us trying to make vaccines against retroviruses is that reverse transcriptase is so bad at what it does that it's constantly creating mutations. And so each virus that's being produced, not each virus, but many viruses that are being produced are genetically different. They're constantly mutating. So making a vaccine is much more difficult for a virus like HIV, which is a retrovirus. We've been studying HIV since the 1980s, and we still don't have a really good vaccine for HIV. We've got some that are on their way, but they're still not effective. All right, moving on. Number 6, explain how the binding of transcription factors to promoter regions affects gene expression. So that is, we already talked about that, but I'm going to back it up just a little bit. Give me a moment. That is, these are so awkward sometimes. Right here, that's what we're talking about. So the transcription factors binding to the DNA and allowing the RNA polymerase to glom on. Okay, promoters are DNA sequences upstream of the transcription start site. They always start with the Tata box in a eukaryote. And then this part is what relates to gene expression in prokaryotes and eukaryotes. Negative regulatory molecules. We're talking about negative control, negative feedback, inhibit gene expression by binding to DNA and blocking transcription. So an example of that, I added this isn't in the College Board, the repressor that grabs onto the lac operon, that's a negative regulator. There are also repressors that can bind to enhancers, and I wanted to show you that to remind you. Give me just a second to think where that is. That's this section right here. Bring it up close. So here we saw the diagram had activators, which actually trigger transcription, but you can have repressors here, which negatively control and prevent transcription from happening. So it's possible to have that in a eukaryotic cell. And then CAP is a positive regulator in the lac operon. They're talking about negative ones here, but it is a positive one. I think I'm just going to keep barreling through on this learning targets video, you guys. It might be a long one. You'll just have to watch this in chunks. Number 7, explain the connection between the regulation of gene expression and phenotypic differences. So remember, again, what they're talking about is the gene expression. So like the proteins that are being made, my eye color being green or brown. Gene regulation results in differential gene expression. So that's talking about turning the genes on and off. That's what gene regulation is. And so we have, if our genes are turned off, we have less of a particular protein than if our genes are turned on, we have more of a particular protein. In the lac operon, if there's no lactose present, then we have the gene turned off. It's not in humans. They have the gene turned off, and there is less of the enzymes to digest lactose. But if lactose is present, the gene gets turned on, and we have lots of the gene for digesting lactose, lots of the proteins, for digesting lactose. All right, certain small RNA molecules have roles in regulating gene expression. They write it in one sentence, and it is phenomenally complicated. I will just mention the one time that we saw small RNAs was on number 4 of this presentation here. So we had 1, 2, 3. This is how eukaryotes have control over their genes, and it's number 4. The different types of small RNA, they often do what's called RNA interference. So they can be called microRNA, they can be called miRNA, they can be called RNAi. So many synonyms for the same thing. But the basic idea, let's see if I can show you this image here. They are double-stranded RNAs that get cut up by an enzyme that's called dicer, and then they bond with proteins. You don't really need to know the details. What's important about RNA interference or small RNAs is that they degrade mRNA. So if they bond so they have complementary sequence with a particular mRNA, they will cut it up into pieces. And so whatever that mRNA was coding for, it's gone now. So that's a way of controlling the gene, is by getting rid of the mRNA. Another thing that the interfering RNAs can do is just block transcription. So right now, this mRNA can't make it through a ribosome, so the translation has been blocked. Even if it doesn't get cut up, it can't fit in the ribosome, so it's never going to get translated. So that is another way of controlling gene expression. All right, number 8, describe the various types of mutation. Let's see, that's in, give me a second. I think that's in this one. Give me a moment. So remember, we have nonsense, we have missense, and we have silent mutations. And then we also have frame shift mutations. So we've been talking in this unit about point mutations, which are teeny tiny little changes. Maybe one letter is wrong or a couple letters are wrong. So point mutations are silent if usually due to wobble, which means if the wrong letter is on the third letter of the codon, then oftentimes it still codes for the same amino acid, and so it's a silent mutation. It has no impact. Something that is similar to a silent mutation but isn't considered officially a silent mutation is if the third letter changes and it causes a new amino acid that is similar biochemically, so the protein doesn't change significantly because it's a similar amino acid, so that a person who has that mutation, even though it's a different amino acid, they might get away, their protein might still function. So is that silent? Kind of, but not officially. All right, and then there's the missense. That's where we actually have the wrong amino acid, and that is especially a problem if the amino acid is different biochemically. And then nonsense. Stop your nonsense, ladies and gentlemen. Just stop. So that's where the change, the mutation in the DNA actually causes a stop codon to be there prematurely. So you should be thinking about if the mutation happens early in the mRNA or DNA, or if it happens later in the DNA, what impact that has on the protein. And then we talked about frame shift mutations. And remember, a frame is the groups of 3 letters, the codons. And anything that screws up those groupings of 3, the ribosome doesn't know any better. It just reads 3 letters at a time. So the 2 things that screw up those groupings are insertions and deletions. That screws up the groups of 3. So an extensive missense is where everything after the insertion or deletion gets screwed up. Immediate nonsense is where the screw up leads to a stop sign again, stop your nonsense. And then the last one, limited effect, is where the insertion or deletion actually occurred in a group of 3. And so maybe an entire amino acid is skipped or an entire amino acid is added. But it doesn't screw up everything after the insertion or deletion. And so it might be terrible, but it might not have as big an effect as a mutation that happened, a frameshift mutation that happened earlier in the DNA. All right, so changes in genotype result in changes in phenotype. So as we get mutations, my eye color changes as I have mutations in the gene that produces the protein that is in my eye. All right, disrupts genes, causes new phenotypes. Alterations. Okay, alterations in DNA sequence can lead to changes in the type or amount of protein produced and the consequent phenotype. So I could produce too much of the protein in my eye if there's a mutation, or not enough. DNA mutations can be positive, so they can give me an advantage, like X-ray vision. Okay, not really, but it could. We looked in the rock pocket mice and we learned that a mutation can give them a color advantage that allowed them to be camouflaged. It could be negative if it leads to too much protein in my eyeball and I go blind, or it could be neutral, meaning it didn't give me an advantage or a disadvantage. Moving on, explain how changes in genotype. Okay, again, this is the part that I was saying was very repetitive. Genotype and phenotype. Errors in DNA replication, that's where we get mutations, you guys, is when during mitosis, the cell is trying to copy the DNA. So that's replication. Sometimes it makes mistakes. We have proofreading enzymes that come through and try to fix that, but we still have some mistakes, and those start to accrue over time, and we learned that in a previous unit. So things that can cause that: DNA replication, errors in DNA replication, and the repair mechanisms not working correctly. External factors like radiation, ultraviolet radiation can cause thymine dimers, where the thymines bond with each other and create a bump in the DNA. Or nuclear radiation, God forbid, creates enormous mutations throughout the body, not just in the skin like ultraviolet radiation. Even exposure to chemicals. Cigarettes, for example, are full of chemicals that create mutations in our DNA. So whether a mutation is detrimental, beneficial, or neutral depends on the environmental context. So with the rock pocket mice, if they were living in light-colored sand, then it's an advantage to be light-colored, and the mutation is a disadvantage, and they're more likely to be captured by predators. flip that around, if they're living on the lava rock, which is dark, the dark mutation is an advantage, and the light colored ones are more likely to go extinct because they get eaten by predators. So it just depends on the environment whether a mutation is good or bad. Mutations are the primary source of genetic variation. Errors in mitosis and meiosis can result in changes in phenotype. This is one we already covered in our last unit. It just comes back again. Changes in chromosome number can lead to triploidy or polyploidy. Sometimes that leads to sterility. Definitely in vertebrates, triploidy is fatal, 100%. Even having one extra, which is trisomy, it has a syndrome associated with it. Some organisms are normal if they are triploid or polyploid, and that can be an advantage, because if one of the chromosomes has mutations, it's got a deleted section, it's got an inverted section, the other chromosomes can act as backup copies and can protect the organism from mutations. And then you should know Down syndrome is trisomy 21. Turner syndrome is an X, a female who has one X and no other X chromosome. We're getting close, you guys. There's two more. Explain how alterations in DNA sequences contribute to variation that can be subject to natural selection. So that's like the rock pocket mice. The changes in their genome, their mutations led to phenotype differences, which can get impacted by natural selection, which ones survive to have babies. So survival and reproduction gets impacted by that. This is one that gets kind of squeezed in this unit, and so I had to squeeze it in at the end. The horizontal acquisition of genetic information. So horizontal transfer, that's what prokaryotes do. There's other organisms that have horizontal transfer also. We do not, generally. There's one exception to that. So how do prokaryotes evolve? How do they change? Because they're cloning themselves. They just go through what's called binary fission, which is basically mitosis, and they're making identical clones. How do they ever change? I think my dog's about to bark, so just brace yourselves. I'm just going to go right through this. So there are 4 ways that bacteria can change. Transformation is this amazing ability that they can just suck up DNA from the environment. My dog is growling in the background, sorry. Trixie, stop. They can suck up DNA, and whatever DNA they suck up, they now have those characteristics. That's called transformation. We can use that now. Now that we know bacteria can do that, we can now take genetically engineered plasmids, put them in the environment of a bacteria, and trigger it to pull up that plasmid, and then we can see what happens. What does that bacteria do with that new piece of DNA? And it helps us to study genetic information. So then there's also conjugation, which is kind of like bacteria sex. This again usually has to do with the plasmids, which are the small circular DNA sections that they have. It gets copied and then transferred, and it can be to a different species of bacteria. So it's like if we could give human DNA to a bird or something, like bacteria can do that. We can't do that. The next way that bacteria can get new information is through transduction. I think this is the hardest to remember one. This is where bacteriophages, little viruses, accidentally pick up some bacterial DNA from their first host. And then when they go to infect the next host, they transfer some of that bacterial DNA. And so now this original bacteria's DNA is now in a new bacteria. So we've horizontally transferred DNA. It didn't happen through sex. That's usually how DNA gets transferred from parent to offspring. And then the last way that bacteria do it is called transposition. Transposition has to do with very bizarre forms of DNA that are capable of moving. So sections, entire section, and this is true in humans also. We have things that are called transposons, but they're commonly called jumping genes. And that means that this is entire sections of DNA that can leave one chromosome and move to another chromosome. The important example of that we gave was in this particular bacterial. So this is the bacteria's circular chromosome. So that's the big section of DNA. In this case, the transposon is an antibiotic resistant gene. It moves to the plasmid, and now now it's in the plasmid. And the reason that's important is that now if that bacteria participates in conjugation, it will transfer that antibiotic resistant gene to a whole new species of bacteria, not just to its offspring, but to a whole new species of bacteria. And this is how antibiotic resistance is spreading all over our bacteria. All right, moving on. No, this is getting long, you guys. Reproduction processes that increase genetic variation are evolutionarily conserved. That means that evolution likes genetic variation, and we'll learn about that more in the evolution unit. Okay, we have not talked about genetic engineering. This is the very last section. So I want you to be really clear. Look at this exclusion statement. The details of these processes are beyond the scope of this course. You would need to take a biotechnology course. The focus should be on the conceptual understanding of the application of these techniques. So you don't necessarily need to know how they work. You need to understand why we would use them and what's important and how they could be used. So the first one that we talked about was electrophoresis, or one of the first ones we talked about was electrophoresis. And I'm trying to remember if I have a picture of electrophoresis somewhere. Give me a second. Okay, so I sort of have that here. So with electrophoresis, remember the idea is that you have a strand, you have DNA, and we add these enzymes that are called restriction enzymes that cut the DNA into pieces, very specific pieces. Then we put the DNA, and I'll see if I can get you an image here. Then we put the DNA into an electrophoresis machine. They are negatively charged. So if we zap them with negative, the negative anode of the electricity, it repels the DNA fragments away from it. They spread out. The smaller fragments travel further. The bigger fragments can't travel as far. And so you create what is sometimes referred to as a DNA fingerprint or an electrophoresis image. And that's what you have right here. So what you need to know isn't necessarily how they do the DNA fingerprint, but why would we use that? So DNA fingerprinting can determine relatedness. So we use it for paternity testing. We can also tell if organisms are closely related, different species. Chimpanzees would produce very similar DNA fingerprints to humans, but very different DNA fingerprints from a frog or a bacteria, somewhat different fingerprints. Parents share about half of their DNA with their offspring, so you'll see commonalities in their DNA fingerprints. We can also identify the DNA suspects in a crime scene. So be aware that when we're using electrophoresis, it doesn't tell us anything about what the person looks like. We can't even tell their sex. We don't know if they're male or female. We can't tell anything about the person. But if we have a suspect, we can take the suspect's DNA and compare it to the DNA fingerprint from the crime scene, and then we can tell if they match or not. And then the other thing that we used electrophoresis for is identifying mutations. So we can put the wild type, remember that's the normal DNA, in one well, and we can put a mutated version in another well, and we can identify that there's a mutation because they'll produce different DNA fingerprints. All right, PCR. PCR is a way of copying the DNA. I think this is an image of PCR. We have the initial DNA. I'm not going to go into all the details, but you just need to understand that the DNA gets copied and copied and copied. So the application, for example, if you had a tiny, tiny, tiny amount of blood at a crime scene, we could scrape it up, put it in a test tube, add it to the PCR machine, turn the PCR machine on for an hour, and we would have a test tube full of that DNA. Now we can run an electrophoresis on that tiny amount of DNA, and we can make a DNA fingerprint of that suspect. That's just one example. PCR can also be used to copy genes of interest, so genes that scientists are studying. We can make all kinds of copies of it. And then we can use those in genetic engineering experiments, which ties into this last one, or second to last one, bacterial transformation. That's what we're doing in these genetic engineering experiments. So we're going to cut a plasmid open with a restriction enzyme. We have a gene of interest that we're going to insert into the plasmid. And then we know that the bacteria, through transformation, are going to suck up our genetically engineered plasmid. Then we can weed out the ones that didn't suck it up, and then we can study or even milk the ones that did absorb the plasmid for whatever protein they're making. So that's kind of ingenious use of transformation. All right, we're almost done here. DNA sequencing, I don't think we've talked much about that. It's just figuring out the A's, T's, C's, and G's. It's, there's 3 billion letters in a human being. It's incredibly complex. It someday will replace electrophoresis, because electrophoresis just cuts it into chunks. This is going to tell us every single letter of the DNA sequence. So it can be used to determine sequences in differences in nucleotides between a functioning gene and a mutation, or between two alleles. What gives somebody brown eyes and what gives somebody blue eyes? We don't know yet. Someday we'll know the exact letters and why that is. And someday it can be used to replace DNA fingerprinting to ID an individual. Whew. Okay, ladies and gentlemen, that's it. I'm sorry it was so long. I hope that you sped it up. Have a great rest of your day.